So here we're going to look at another hallmark of cancer, which is on genome instability and mutation. Now, there are some key points listed here which we're going to cover. The first one being DNA repair deficiencies. So in every cell we have, so this is your cell, and then you have your nucleus, and inside your nucleus lies your DNA or your genomic material. And if there are mutations in the DNA, there usually are mechanisms to fix it. So this is a tick mark. So there are mechanisms within the cell and the nucleus to prevent and to repair any damage to DNA that occurs. However, when there is deficiency in the repair mechanism, so there are repair mechanisms, and there are many different types of repair which are in response to many different types of DNA damage. So DNA damage leads to repair mechanisms. So and there are many different types of DNA damage, many different types of repair. Now, if this repair is somehow mutated and is non-functional or malfunctional, so it could be either malfunctional or non-functional. And again, this is a very simplistic here, right? Explanation of what actually goes on. So again, many different types of repair mechanism, and each repair mechanism has again many different components, and say the malfunctioning of one component can lead to downstream effects, right? And this can lead to genome instability and mutation. So if a mutation occurs and then we have this specific malfunction or non-functional component of a DNA repair mechanism, then we have this mutation still existing within the genome. And depending on the type of the mutation, so there are many different mutation types, right, or DNA damage types, and depending also on the specific gene that is mutated, there can be various different outcomes. So various outcomes to the cell as a result of a DNA damage that was not repaired due to a malfunction or non-functional component of a DNA repair pathway. So DNA repair deficiencies or mutations can lead to genome instability and mutation, which is the hallmark, right, of cancer. Now let's look at the second point here, epigenetic changes. So just to go back to our first point, changes made to DNA, such as a mutation as indicated by the line here, are genetic changes. So genetic changes. But sometimes we hear the term epigenetic changes. Now, to understand this, we need to know what epigenetic, epi stands for and what it means, right? Well, the DNA is bound by proteins. Now, again, this is a very simplistic illustration of what actually is. And these proteins are involved in the modulation of the shape of DNA such that it either allows genes to be expressed or not expressed. So you can have an analogy of something that is tightly coiled, and the gene that actually resides here is inaccessible to be expressed, whereas your DNA, if it's free and there are no proteins sort of surrounding it, then it is going to be accessible for expression in terms of having the gene being expressed and produce some sort of protein, the protein inside the cytoplasm. But not to get into too much detail, though, there is both genetic changes and epigenetic changes. And epigenetic refers to the modulation of the genetic expression via proteins. So this is via proteins. Now let's look at the third point here, which then again contributes to genome instability and mutation, and that is related to replication stress. So DNA, as we had mentioned, is when dividing, right, we have one strand, parental strand here, one parental strand for the other daughter cell, and a new strand, right? And these are again within the nucleus of the two daughter cells. Now what we need to sort of understand is that during this process of DNA replication, which is the copying basically of the genetic material of the DNA, there can be processes, important processes that create stress. And there's a whole bunch of protein machinery that sort of allow the replication process to occur. And if this replication machinery is defective or non-functional or malfunctional, right, any of those things, or then there can be replication stress, which then contributes to the mutation in the genome as here. Then we have oncogene activation. Now oncogenes are genes that, when active, are going to lead to proliferation of the cells. So if you have, making some space here, if you have one original cell, and if you have oncogenes super active, then this cell is going to divide and divide and divide, right? And there are many different genes, right? So within your genetic material, within your DNA, there are many different genes in there that, once hyperactive, then it leads to phenotypes such as being highly proliferative. So dividing, right? And usually this phenotype is seen with stem cells. Stem cells have this sort of proliferative phenotype. Now, looking at the last point here, which is tumor suppressor inactivation. So if this was oncogenes, and there are other gene types that are referred to as tumor suppressors. So you have the genes that if you have them mutated, then it will lead to this proliferation. So opposed to oncogenes, which once they are hyperactive, then you have this proliferative phenotype. In case of tumor suppressors, once they are inactivated via DNA damage mechanisms or types, then you have this sort of proliferative phenotype. So these are some of the mechanisms by which genome instability, so the genome here, and mutation occurs, and there are various different mechanisms in each of these, and perhaps more points here, and each of these points sort of has their own detail, but here we've gone over some main sort of general themes that we see in the context of genome instability in cancer.